All right, class. So now we're in chapter 4, a tour of the cell. Again, lots of information to get through in these chapters. So I'm going to focus on kind of the key components that I want you guys to focus on and what I'll be kind of questioning you in. Also use those guided readings to take you through these chapters as well. All right, so as we know, cells are kind of the unique part of life because cells are what will make up every living organism. We have two major categories of cells, and I'll start here. Biologists can classify all life into three major groups called domains. We have prokaryotic cells and eukaryotic cells, and these are some differences between the two. Prokaryotic cells, you can see, are smaller, simpler, found in bacteria, archaea. Most have cell walls, some capsules. They have a plasma membrane, but no membrane-bound organelles. The eukaryotic cells are much more complex. They're found in protists, animals, plants with a plasma membrane and with membrane-bound organelles. So here's a look at a prokaryotic cell, much simpler, no organelles. They do have ribosomes, which synthesize proteins, and usually a capsule or a covering. And here's an idealized animal cell and a plant cell. These are eukaryotic cells, kind of showing their plasma membrane, their organelles inside, and so forth. Here's an idealized animal cell. What you're looking at is the plasma membrane surrounding it and all of the organelles inside of it, which are like all of the machines, the kind of the machinery that help that cell perform its function. Please take a look at the tour of an animal cell and the tour of a plant cell. Those will be extremely helpful. So the plasma membrane is what separates the living cell from the non-living surroundings. It's made up of a two-layer sheet called a phospholipid bilayer, and we call the plasma membrane a fluid mosaic kind of a fluid mosaic model because molecules can move freely past one another, and it's a mosaic because of the diversity of proteins in the membrane. So here we have kind of the lipid bilayer of a membrane and the fluid mosaic model showing the embedded proteins and how structures will freely move through it. Plant cells have a cell wall made of cellulose fibers, and animal cells lack cell walls and most secrete a sticky coat called the extracellular matrix. All right, I'm going to keep going forward. You can read through that a little bit on your own. The nucleus is kind of the main command center inside the cells. It has long DNA molecules and associated proteins that form chromatin, and the nucleus is the site where the components of ribosomes are made. So there we see the nucleus, kind of the main central player in the cell. And here are the DNA surrounding proteins making chromatin fibers and eventually forming the chromosomes, the genetic material. Ribosomes are then responsible for protein synthesis. In eukaryotic cells, components of ribosomes are made in the nucleus and then transported out into the cytoplasm where they work. So there's a look at a ribosome and how it's going to create a protein coming out of it after reading an mRNA instruction structure. Here's our endoplasmic reticulum-bound ribosomes. So some ribosomes are attached to another organelle called the endoplasmic reticulum, and they are shown as those tiny dark blue dots. How does DNA direct protein production? So DNA transfers its coded information to a molecule called messenger RNA, which then leaves the nucleus, travels to the cytoplasm, binds to a ribosome, and at the ribosome, as a ribosome moves along, the messenger RNA kind of like reading it and translating the genetic sequence message into a protein with a very specific amino acid sequence. So DNA is created. It's kind of what we call transcribed into an mRNA structure, which will leave the nucleus and then bind to a ribosome. The ribosome reads the messenger RNA and then translates it into a protein. The endomembrane system consists of the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, and vacuoles. And all of these membranous organelles are either physically connected or linked by vesicles. So the endoplasmic reticulum produces an enormous variety of molecules. It's connected to the nuclear envelope, and it's composed of interconnected rough and smooth ER that have different structure and functions. So the rough ER is kind of embedded with ribosomes, and the smooth ER does not have any ribosomes. So the rough ER makes more membrane. It refers to the ribosomes that stud it. And these ribosomes attached to the rough ER produce proteins that will be inserted into the growing ER membrane or transported to other organelles. So you can see that there. The smooth ER does not have ribosomes. It produces lipids like steroids and helps the liver cells detoxify circulating drugs. The Golgi apparatus works in partnership with the ER. It's kind of like your cell's post office. It receives, refines, stores, and then ships out or distributes chemical products of the cell. You can see that Golgi apparatus shown there. Lysosomes, then, these are membrane enclosed sacs of digestive enzymes found in animal cells. Most plant cells do not contain lysosomes, and enzymes in a lysosome can break down large molecules like proteins, polysaccharides, fats, and nucleic acids. They can also destroy harmful bacteria, engulf and digest parts of another organelle, and then sculpt tissues during embryonic development to form structures as fingers. Those are lysosomes. Vacuoles are large vesicles. Food vacuoles will bud from the plasma membrane, and certain freshwater protists have contractile vacuoles that pump out excess water that flow into the cell from the outside environment. You can see these two types of vacuoles as shown there. All right, this is a little review of your endomembrane system. Chloroplasts are a part of photosynthesis. They're unique to photosynthetic cells of plants and algae, and these are the organelles that perform photosynthesis. Really important to remember that. That will probably be a test question. So the chloroplast is the site of photosynthesis. Mitochondria are found in almost all eukaryotic cells. These are the organelles that produce ATP from the energy from the food molecules it intakes. You can see there's a mitochondria. Cellular respiration is the process of turning glucose into ATP. Chloroplasts and mitochondria contain their own DNA that encode for some of their own proteins. And then the cytoskeleton. This is a network of protein fibers extending throughout the cytoplasm. It serves as both the skeleton and muscles of the cell, and the cytoskeleton will provide mechanical support to the cell and help a cell maintain its shape. We have microtubules. We have cilia and flagella. Flagella will propel the cell kind of in a whip-like motion. The only human cell that has a flagella is the sperm cell. Cilia are shorter and they move in a coordinated fashion. They also propel protists through water, and cilia may extend from non-moving cells. For example, in the human trachea, cilia help to sweep mucus with trapped debris out of the lungs. So there's a look at flagella and cilia lining the respiratory tract. And please look through those other videos as well. And I think that's all we get through in this chapter. So these are shorter lecture PowerPoints because I know there's a lot of information for you guys to get through on your own. And I will try my lecture videos to focus on the important